When you think about wastewater treatment, your mind probably doesn't jump to, cutting-edge biotech, but it should. Because hidden inside these facilities is this incredible, bustling world of trillions of microorganisms. And today, we're diving into how scientists are becoming architects of this microscopic world. How they're actually training bacteria to build their own tiny, super efficient water-cleaning cities.
So that brings us to the big question, right? How in the world do you get trillions of tiny individual bacteria to cooperate? To get them to stop just floating around doing their own thing and start building these perfect, dense little spheres together? I mean, we're not talking about a small petri dish here. This is a massive engineering puzzle.
All right, so to really get this, we first have to understand the old way of doing things. For about the last 100 years, the go-to method for cleaning wastewater has relied on something called activated sludge. And look, it gets the job done, but it's got one major, fluffy problem.
And you can see exactly what I mean right here. On the left, that's your conventional sludge. It's basically a loose, messy, fluffy cloud of bacteria. And because it's so lightweight and all over the place, it takes forever to settle, which just slows down the entire process. The goal, what we're aiming for, is on the right, the complete opposite. We want dense, compact, spherical communities that settle out fast.
Now, if you want to put a number on that inefficiency, scientists use this metric called the Sludge Volume Index, or SVI. You can just think of it as a fluffiness score. The sludge they started with in these experiments, it had an SVI of 208 milliliters per gram. And trust me, that is a huge number. It basically means the sludge is taking up a ton of space for the amount of work it's actually doing.
So yeah, fluffy sludge is a problem. Clearly, there has to be a better way. And that better way is to get the bacteria to build what you can really think of as these perfectly formed microbial cities. In the science world, they're called aerobic granules.
And this is what they look like. It's such a huge difference. Unlike those loose bits of sludge, aerobic granules are these tight, spherical powerhouses of microorganisms. They're heavy, they're dense, and because of that amazing structure, they settle like a rock, which makes the whole treatment process way, way more efficient.
So, how do we get them to do this? What's the secret? Well, it's not some magic chemical you pour in. It's actually a really clever training program. It's basically a boot camp for microbes, where you create a set of environmental pressures that forces them to adapt and work together.
The core idea here is something called selection pressure. You can think of it like evolution on hyperspeed. You create this really tough environment where only the microbes that figure out how to band together and form these dense, fast-settling granules get to survive and stick around. It's like a tryout, and if you can't form a tight, heavy team, you literally get washed out of the system.
And this training program has three main hurdles. First up, a really short settling time. You give the microbes just a few minutes to sink. Anything that's still floating around, all that fluffy stuff, gets washed away. Second, you hit them with high shear force. Imagine a really strong current that's constantly trying to rip them apart. This forces them to build stronger, more compact structures just to survive. And third, you have these feast-famine cycles, which messes with their metabolism and encourages them to store food, which also helps them clump together.
Okay, so that's the what of the training program. But how does all that pressure actually make individual bacteria want to stick together at a microscopic level? Well, it all comes down to two really cool biological changes.
The whole thing kicks off with a change in the bacteria's relationship with water. Under all this stress, their cell surfaces become more hydrophobic, which is just a fancy way of saying they become water-fearing. And just like little droplets of oil in water, these hydrophobic cells realize it's way easier to just clump together to minimize how much they have to touch the water. That's the initial trigger. That's the first handshake.
And you don't have to take my word for it. The data is right here. The original sludge started at about 49% hydrophobicity. But after going through the training program, the finished aerobic granules shot up to over 81%. This is a huge fundamental change in their chemistry, and it's what gets the whole party started.
So once the bacteria start huddling up, they need a way to make it permanent. They start secreting this stuff called extracellular polymeric substances, or EPS. And you can just think of this as a biological superglue. It's the sticky web of sugars and proteins that weaves through the community, binding all the cells together and giving that tiny microbial city its incredibly strong and stable structure. So we've got the theory, right? You apply pressure, you trigger this water-fearing behavior, and then they produce their own biological glue. So what does that actually look like in practice? Well, the results are nothing short of a stunning transformation. Check this out. After just 1 week, you can already see the first little clumps forming. By week 2, you've got these early-stage granules appearing. And then, after only 3 weeks, the whole system is dominated by these beautiful, mature, perfectly round granules. It's amazing. A chaotic, fluffy mess becomes an ordered, highly efficient system in less than a month.
And here it is. This is the big payoff. Remember that fluffiness score we talked about at the beginning? That crazy high SVI of 208? Well, after this process, it plummets all the way down to just 36. That is a massive improvement. It means the new granular sludge is incredibly dense, and it settles out of the water in a tiny fraction of the time.
Now, this is more than just a cool science experiment. This has huge benefits for the real world. It means new treatment plants can be built on a much smaller piece of land. They can handle more waste, and they can operate way more efficiently. This tech even gives us a way to upgrade older facilities without having to do massive, expensive new construction.
There's this quote from the research that just sums it all up perfectly: inducible rather than constitutive. In simple terms, what that means is that bacteria don't normally do this. Forming these perfect granules isn't their default state. It's a behavior that we can switch on, that we can induce through really clever environmental engineering. We are actively guiding their evolution to get them to do what we need. And that just leaves you with this incredible, mind-bending thought. We've learned how to be city planners for bacteria just to clean our water. We've proven that we can shape entire microbial communities to do these really complex, useful jobs for us. So what's next? Can we train them to clean up oil spills, to break down plastics in the ocean? The possibilities of what we can teach these tiny engineers to build, well, they're really just beginning to unfold.